Okay, so in the next 15 minutes, I'm going to try to introduce a new approach to complex disease subtyping using contrastive learning. It might be comforting to view each disease as a homogeneous monolith given the diagnostic criteria laid out in modern medicine. But a closer inspection often reveals that there's a lot of underlying heterogeneity. So we'd like to classify patients of a disease into more clinically homogeneous subgroups or subtypes, because this allows us a better mechanistic understanding of the heterogeneity in the disease, which could help us derive better treatment outcomes and more targeted prevention strategies. And this becomes clear when you consider canonical subtypes such as type 1 and type 2 diabetes, ER positive and negative breast cancer, responsive versus non-responsive bipolar disorder, right? Implicit in these examples is the fact that we're interested in investigating signals or heterogeneity that's within cases, so that we can learn heterogeneity of the mechanism of the disease. So let's consider an illustrative toy example where we have a bunch of cases from a disease, and I've told you that there are two subtypes, and the subtypes are given by the colors, the red and the blue dots. But what I didn't tell you is that there are two confounders, A and B, right? If you visualize this data in higher dimensions, you can see that most of the variation in the cases comes from these confounders. But if I don't tell you that these are confounders beforehand, and you run classical dimensionality reduction or clustering approaches, you get something like this. And what you're essentially doing here is you're tagging the confounder. But if you don't know that this is a confounder beforehand, you're going to spend days or weeks trying to figure out what this subtype is, only to find out it's something like library preparation. So what you'd actually like to do is find the weaker axes of variation in the case data that is unique to the cases, and if you can do that successfully, you should be able to remove this shared confounder between the cases and controls and get a corrected version of the case data where you can then just isolate the subtype using standard dimensionality reduction or other approaches, right? Okay, so one way to do this is by using a contrastive approach. When trying to find subtypes in the cases, we actually have extra information that's in the controls that we can use to subtype the cases. And what I mean by that is that the confounders, if they're not case-specific or disease-specific, are shared with the control population. So, in theory, we should be able to contrast this disease and this without disease control data set and isolate out all the shared confounders removed from the case data set. And if we do that properly, we should be able to isolate the case-specific variation, so find subtypes 1 and 2. And this is exactly what we try to do with PACA. So PACA stands for Phenotype Aware Components Analysis, and let's specify a model under which PACA works. One, consider controls of the form like M by N, so it's features by samples, and this blue matrix here is just, it defines some subspace or factor or directions, right? And this yellow thing here defines the structure in that subspace. Now, if you can do the same factorization for the cases, but now if you assume that the blue matrix here is shared between the cases and controls, what that essentially represents is the direction of shared variation between the cases and controls. So that means this term here is the shared signal between the cases and controls, and the second term here is the case-specific signal. Now, if you make an additional assumption that the blue thing here and the red thing here are orthogonal, you can just easily isolate the shared signal and subtract that from the cases to isolate this case-specific signal. And that's exactly the objective function that PACA tries to solve. It tries to identify what this maximal direction of shared variation between the case and control are, so this blue matrix. So how do we do that? We use CCA to do that. And it's important to keep in mind that normal CCA tries to solve for settings where you have two different data modalities and shared samples. So when you solve for that CCA objective functions, your A and B, your canonical directions, are weights for the linear combination between features and samples that between features from the two different data sets that maximize the correlation in the sample space. But in PACA, we're actually interested in subtyping, where you have one set of features but two different sample populations. So this X and Y are transposed from your normal input matrix. So they'll be of the form like features by samples, not samples by features. And if you solve for that, what you actually end up getting is an A and B, so canonical directions, that maximize the correlation of the feature space between the two different set of samples. Now, if you do that, your first canonical direction, or canonical covariate, should actually be the strongest direction of shared variation between your cases and your controls, and that results in your first column vector for this blue matrix that you're interested in estimating. Now, you can keep doing that iteratively to find all the case strongest directions of shared variation between your cases and controls, and voila, you've estimated your shared component. But there is an issue, right? Generally, the rank of this blue thing is unknown, but PACA also solves for that automatically, and the way we do that is we try to choose the K, the minimum K at which there's only significant variation in the case data set, in the corrected case data set, and not in the corrected control data set. And we can take that intuition and incorporate it into a permutation testing framework to estimate this K. Now, I've given you a high-level intuition for what PACA does and how it works. Feel free to read our preprint for the math and the details behind this approach. So in comparison to existing methods like contrastive PCA or CPCA, we don't have to estimate an ambiguous hyperparameter. While in contrastive PCA, you're essentially going to have to manually select this and do some sort of grid search to find your optimal hyperparameter, which you don't know beforehand and might be hard or impossible to choose without some information about the kind of subtype you're trying to find in your data. In addition, it's also insufficiently powered and not calibrated depending on the setting, right? And I can actually just show you this by considering some real data examples. So consider some methylation data where we take a mixture of cases, like cases, where they're all Mexican and Puerto Rican ancestry individuals, and the controls are Europeans. And note that this data set comes from a single tissue, so there shouldn't be any significant tissue type variability. And the only difference between the case and the controls are the population structure. So, oops. So a well-powered contrastive subtyping approach should be able to distinguish the Mexican versus Puerto Rican ancestry samples. Now, obviously PCA failed because this does not account for like the variation in the controls, and it's probably just tagging something like age or cell type composition. But interestingly, CPCA fails as well, and this is after doing a grid search of all possible values, and we find that the best results from CPCA, you get the best results from CPCA when you like set the hyperparameter to something close to infinity, so like 1,000,000 or 1,000,000,000, right? But PACA, on the other hand, does show stratification between the two subpopulations, and it does so while automatically learning that it needs to isolate the top 11 shared axes of variation. Next, we can also consider cases where there is no subtype signal. So this is an important feature for subtyping methods because you don't want to be wondering if you've just tagged a population confounder or like it's something that is case specific. So to test this, we can consider some European and African ancestry samples from the HapMap data. There are no true cases here, so we just randomly assign the samples to cases and controls. And a good subtyping method that's well calibrated should show no separation between any samples. So as expected, PCA tags population structure. Everyone here would expect that. But interestingly, CPCA also tags the same population structure, but it's just slightly less obvious. PACA, on the other hand, does remove all shared variation between these cases and controls and results in no stratification. And to be even more exact, PACA also has a consistently calibrated type 1 error rate, and there's like results in the paper if you're interested, in not just genetics, but also gene expression. Next, we can go even further and consider some gene expression data from psych and code, where we actually have cases, and the cases are consistent of schizophrenia and bipolar disorder patients, and the controls are healthy. So a well-powered subtyping method should really be able to differentiate between the schizophrenia and bipolar disorder samples. But if you look at the PCA decomposition of the data, you can see that all the cases are perfectly separated by library prep type. So additionally, a good contrastive learning method should also be able to capture something more than library prep, even though library prep enriches for like schizophrenia in this case. Unfortunately, CPCA does not do this. Like contrastive PC1 is not significant after you correct for library prep, but PACA is. So we see a significant association with the case subtype status for PACA, which survives correction for library prep and the first couple of PCs. So we think this is an important result because a good subtyping method should not just be restricted to one or two modalities, because there's no guarantee that any given subtype will exist for all diseases in the same data modality. And some of you who worked in this know that to be the case. And we think this is a key advantage that PACA provides because it's a general framework for subtyping, not just for like one data set. So finally, we can go ahead and apply PACA to some real data where we don't actually know if there are subtypes, and if they do exist, what they might be. So we have access to this large study called Converge. Converge has about 7,000 samples of recurrent MDD cases and controls, exclusively from mainland China, and they're all females. It's unique in the sense that it has deep phenotyping, so they have a lot of information about all the, a lot of questionnaires, family history information, and stuff like that. But it also has whole genome sequencing, and most importantly, we have voice data for all the participants in the study and an independent replication data set of MDD cases and controls from the same population collected 10 years apart. So what we're going to try to do now is try to see if we can find subtypes of depression using voice data, and if we can replicate this in the independent study. So we first apply PACA to the Converge Discovery voice data set, and we're actually able to find PACA PC1 is significantly associated with and driven by education, previous treatment type. This is whether you've had medication or medication and therapy before, and menopausal status. And we were actually able to replicate the education signal in the independent replication data set. Unfortunately, we couldn't replicate the previous treatment data type because that phenotype just doesn't exist in the replication data set. But we went ahead and actually also did a GWAS where we found a SNP that survives multiple testing correction, and it's on the Cas-N gene, and this gene is interesting because there have been a lot of studies that have shown SNPs on this very gene that associate with educational attainment, treatment resistance to MDD, suicidal thoughts, and other depression phenotypes. So this work is still ongoing, and we hope to see if there's like some important biological implications in relation to PACA PC1 and subtypes of depression. So, in summary, PACA is a novel contrastive subtyping approach, which, unlike existing methods, doesn't need hyperparameter tuning, is calibrated, and well-powered for biological data subtyping. We were also able to successfully recapture axes of variation unique to cases and reveal disease subtypes in positive controls. And it's a general framework for model-based contrastive learning, which is not data type agnostic. And I haven't mentioned this, but you're not actually restricted to one data type. You can go ahead and combine something like voice data and gene expression data, right? Like there's no restriction on what that feature set is as long as they're shared between the cases and controls. And finally, we're also able to show that PACA finds a putative and replicable subtype in voice data, which is being further tested for implications in depression subtypes. So yeah. I'd also like to thank a lot of people, Elior, who originally came up with this idea, my advisors, Jonathan and Sriram, who've been very supportive and helpful, and then Noah, who's during my rotation with whom I started this project, and a lot of people in the voice depression subtyping working group who've helped process and clean this data, and obviously the NIH for funding this whole thing. Yeah, feel free to contact me on this email or check out our preprint using that QR code, and I'm happy to take any questions.